Hello, everybody, and welcome to 17 Minutes of Science. This is In Vivo Biosystems show, where we interview scientists and thought leaders who have something to tell us about science. And today I am very excited to be talking to Alex Parker. Alex Parker is an associate professor in the Department of Neuroscience at the University of Montreal, and he is a researcher at CR Schum. Alex is also a medical geneticist by training, and he has an interest in late onset neurodegenerative diseases. He uses the model organism C. elegans in combination with mice to confirm his findings. A major focus of his successful academic career has been the development of models of ALS, or Lou Gehrig's disease. These studies have led the way to identify new therapeutic targets and strategies directly usable in the patients, and among them, a drug has been discovered in his lab that is proven successful in a clinical trial for ALS. So welcome, Alex.
Oh, thanks a lot. Thanks for having me. It's a fun idea. I'm happy to be here and talk a little bit today. So thanks.
Wonderful. Yeah. So my first question is just pretty broad, but how about you tell us about your research and what's going on in your lab right now?
Yeah, so I work with worms. I've worked with C. elegans my whole career since being a grad student to postdoc to like PI now for a while now, I guess like 13 years. And I've always been interested in neurodegenerative diseases. I don't know why. It was just I just got into that field, and all along I've been trying to use worms to make models. And from there, really, we spent a lot of time making all kinds of different models. Like in my lab now, the main focus is ALS, but we've been developing models more broadly for neuromuscular disorders, as well as we're branching into things like dementia and whatnot. And we're really interested in seeing if we could use the worms for drug discovery, because it was like a situation where everybody knew very well that worms are great for modifier screening. Genetic screens obviously are invaluable, and they've discovered so many things, but why not drug screening? So we just thought we'd give it a try, and it turned out to work rather well in the end. So we do a lot of drug screening in parallel. We've probably only done a handful of genetic screens, and like, I don't know, probably over 24 small molecule screens now with the various models.
Wow, that is really incredible. I know that in our work a lot, we got asked about the translatability of C. elegans for human diseases. And one of the things that I really love about your work is that it proves that it really, the worms really do have a lot to say about human disorders, especially with neurodegeneration, it seems.
Yeah, so that's the thing, right? Because you will have to get those comments like, well, what are worms good for? What have you done for us lately? But that's not true. It's like there's a lot of potential still there. And the fact that we were able to relatively recently develop a model in worms, do a drug screen, and with our collaborators move it up through fish, mice, and ultimately to clinical trial means it's possible, right? We're not the only people who are going to do this in history, hopefully. Right. So, I mean, there's a lot of room out there. So it's a proof that you can do this. I think it's just, I don't know what it was. People thought there's not enough translatability. I think it really depends on what your plan for translation is. Like, do you have appropriate or good complementary models to move things beyond the worms? Because if you do, then you can quickly tell if what you're working on is worth it or not. So I think that's it. I think the main thing is that C. elegans is fantastic for discovery, of course. But some of the power is that if you pair it up with other systems, you can move through things quickly.
What has been your strategy in taking the work that you do in C. elegans and moving it through mammalian models?
That's always tough, and it's an ongoing question, because we want to find models that are good for testing therapeutics. And what does this mean? So, I mean, with the technology that we have and the approaches, and with working with worms, you can use huge numbers, right? So you can get the variability down. So there are a lot of good models. Let me put it this way. For any particular disease field, right, I don't know how long it takes for people to develop a consensus as for what is the best mouse model for this disease. It could take decades. And why is that? Because they show phenotypes, but not all the phenotypes are useful for like reliable drug testing. So there are some models that are great, or it's too hard to do. So if we can find a model that shows, for example, most recently, we've been using an ALS model. It's the SOD1 mouse model, the G93A. It's a classic model. Some people say it's a very specific form of ALS. That's fine. But for us, on one hand, it shows reliable neurodegeneration. And that's what we want. We don't want to be is this really working or not? It's like these might show a great phenotype. So that's it. Something that can show reliable phenotypes in a relatively short amount of time. I mean, it's a lot to ask, but and there's not always the best model. There's not always a good disease model for our needs at any given time. So it depends, but we talk about this all the time. And anytime we try to embark on a new project, it's one of the things, what can we do? How far can we actually go? And we're at the mercy of others in this aspect.
Hmm. And what do you think has been the role of CRISPR transgenesis and some of the other genetics methods that are available in worms in creating appropriate genetic models of diseases?
Yeah, so this is great. I've been a big fan of this approach, and I honestly think it is probably will be the standard it'll be the future. I mean, the classic approach of using transgenics is still great. But you know, you're always hindered by sometimes people will say, and they're not wrong, that sometimes there's massive overexpression of your gene of interest will give rise to phenotypes that are not necessarily relevant to what's happening in humans. So it's a trade-off, right? The transgenics are often you have a strong phenotype in a short amount of time, so it's great for screening, but you might be picking up things that are hitting on expression or expression problems. Whereas the CRISPR gene editing models, if you can do that, I believe that's the way to go forward, because you have, you know, it's genetically relevant. But again, you don't know, because that's the thing. Well, with any of these models, I would say probably, let me see, for every model we make, about 60 to 70% of the time we have a phenotype we can use, and some that show nothing, so you have to move on. But I think that if you can gene edit patient relevant mutations in the C. elegans genes, that is the way to go. The trade-off usually is that the phenotypes are not as strong as what you were used to if you're working with transgenics. But you know, you adapt. That's my feeling anyway.
Yeah, you've mentioned a few times that the worms show phenotypes and that they've been good for you in terms of phenotypic-based screens or phenotypic drug discovery.
Yeah.
What do you think is the role for this phenotype drug discovery, PDD, over target-based drug discovery, or as complementary to?
Yeah, so that's the thing, right? So it's a big issue of debate, obviously. And in the end, I'd like to probably say that they're complementary, right? Because if you can narrow down a mechanism and you have, and it's like, you know, I guess you could say, is this mechanism highly penetrant to the disease? I'm not sure you use the terms this way, but then yeah, that's a great mechanism, because you can't get much beyond that. But a lot of the time, we don't have this information. We just don't know yet. I mean, genes will be identified for diseases that are still being identified all the time. And so if you don't have a clear consensus on what the mechanism is, if you're somewhat limited in trying to develop a target-based approach, even for something like ALS, I don't know, there's over 24 genes involved. They're linked to all these different processes. Is there any one key mechanism, or is it likely a mix? So the phenotypic screens, you know, you come in and you're basically, as we like to say, we're somewhat agnostic about the mechanism at this point, because we don't know. So you basically know, what do you know? You know the genetics. This gene, this variant will cause the disease. You put it in your animals and they're sick. Then you go off and do the work. You can do the phenotypic screening. And then once you have a drug that rescues your phenotype in your animals, this is basically a tool at the same time, because then you can go back and drill down on the mechanisms using the genetics and whatever. And I would say that people shouldn't be afraid of phenotypic screening. I think it's actually a very powerful way and can complement or even lead to further like target-based approaches. But you know, we do phenotypic screening basically nonstop, because that's sort of the space we're in, because genes are identified, no main cause is known, but we need to get to work anyway.
Have you ever dovetailed your approaches to phenotype-based drug discovery with later target identification using some of those genetic modifier screens that you were talking about earlier?
We haven't completed that completely. We've done a few things. None of this is in various states of projects, right? I think we've done a couple things where basically you have a situation where the drug rescues, and then you test the candidate genes. I don't believe we've done a classic genetic screen yet to block the effect, right? And then go in and clone it. So that's something I've been bugging people in my lab for a while. And it's just like, it has to be set up right. I think if you can reliably screen that way, that's something we are interested in. We talk about it frequently. It's like, because you have the drug, so yeah, we're just not there yet, but it's something we talk about.
Yeah, I think it does seem like there should be opportunity for that, right? Like that there should be some kind of opportunity to follow up on hits that you find and perform some kind of modifier screens to really pin down the target with C. elegans,
Yeah.
because we do have these tools available. We do have,
yeah. Yeah, I agree. I think that's a powerful way. And there's a handful of papers where people have done this with like a well-known drug. I guess maybe the metformin experiments are good examples of this from the worm literature, where they've identified new mechanisms for what is known to be like a well-known drug. So
right, a very widely used drug.
Yeah.
Including off-label
exactly.
uses for people who want to live to 120. So yeah, I tend to agree with you. I think that's really pretty powerful. So I also wanted to talk to you a little bit about, since you're using patient-specific variants to model diseases, do you think that looking into the future that there is a role for C. elegans, maybe Drosophila and some of the other lower organisms in finding precision medicines that really are specific to particular genetic variants that cause disease?
Yeah, so we do think that, because we think that while some of these genes that exist in pathways maybe all have common pathways, they probably, each specific variant or gene gets there a very specific way. So we're starting to see this where you think that perhaps some of the drugs that you're identifying for one particular gene may be effective overall, but it might be better if you do a second screen for a different variant. So we think you can actually mix and match these up. But I do think there's something there. And some people will say, well, the worm is sick. It's sick. It's really sick because it has too much of this gene or not enough of that. But I think there are differences. So I think this is something that we can. And that's the thing. This is something you could probably do. You could scale this up in C. elegans, because we all probably talk to. Well, I talk to many clinician researchers, and they have bags and bags of variants they need tested. And so they'll talk to somebody like me, and it's like, you can do this, and like, we can't. So we just have to basically set up a more of a pipeline approach. And because you'll hear that certain patients with certain variants they don't respond to certain well-known drugs. So why is that? So perhaps we can screen again against these variants and find something more specific to each patient. So that's something. Yeah, I think there's a huge potential there.
Yeah, that's great. I think that's really good news for the precision medicine community as a whole. You just mentioned you have clinical geneticists knocking on your door of your lab and asking you to take on projects. And I'm wondering, I know that you also have an appointment at Modellus, which is a company that does some of this modeling. Is that related to your founding of Modellus?
Yeah, I mean, it was basically directly related to this, because when we started doing the drug screening, it worked out pretty well. And at that time we started publishing, and when the paper came out showing that with the full story, like the worms up to the clinical trial, we were getting a lot of requests to like try this and try that, but it was becoming too much for my lab to handle in an academic setting. So I thought there was an opportunity there to set this up formally. And that was the birth of Modellus. We're like, why can't we do this? And the company was set up. And so far so good. And we do this all the time now at various levels for various people. Because it was just too much. And I thought we have to do something because it works. I think the approach works.
Mm-hmm. Yeah. Going back to your involvement in this ALS clinical trial, how did you see this progressing, and what was really the timeline of moving from a primary screen and C. elegans all the way to clinical trial?
Okay, so it's getting fuzzy now. Let me see. I think we did something like, I believe it was, and somebody will correct me if I'm wrong, I'm sure. I think from the time we started the screen to the beginning of a clinical trial, like with the recruitment of Lawrence Korngut, basically, and the clinician researcher at the University of Calgary, was 2 years. It was 2 years. Now that's not with everything, right? That's like, we have this, we got to this point to the clinical trial. And in the meantime, we were working out some more of the mechanisms. So it was 2 years, yeah. However, to publish the entire story took like 5 or 7, but that's another thing, so.
Why does publication always lag so far behind?
I don't know. I mean, so something that's interesting, or maybe, something to consider, is that this type of academic drug discovery approach, don't get me wrong, it's great, and you want to translate it as far as you can, for everybody. But I'm not convinced that the system is set up to do this properly for the trainees sometimes, because sometimes by the time, for example, when we talk about this often, is like when our paper came out, all the people who had done the work had moved on, right? So it's a little hard to run these huge things, because it's, it's the thing you're supposed to do. That's what he says. It's like, you want to start with basic discovery with the model organisms and get it to a clinical trial. That's great, but it's hard. It can be long. It can be long for everybody involved.
Mm-hmm.
But that's just something to consider.
There's a recent trend, I guess, towards patient advocacy groups becoming more involved in the pursuit of scientific research. And I'm wondering if you have experience in working with patient advisory groups, and what do you think about their role in moving science and treatment forward?
Yeah, I think it's good. I mean, they organize, and they have their own voice here, and they want to see things get done right, quickly for their families and whatnot, and that this makes sense. And we work with several groups we have, where, for example, there's a very specific, I'm not going to get into details, specific variant with somebody in this group of families or whatnot. And we'll develop models and start to get to work right away. And sometimes this will be funded by these private foundations, and it just moves at a quicker rate than a traditional granting agency. It depends, but this allows us also to develop preliminary data and drug hits that might be interesting to this group, who, in the meantime, well, let me put it this way. Sometimes they'll be like, well, you can develop worms and/or fish model relatively quickly and do a complete drug screen by the time they develop a mouse model. So there's a room here to just get things going quickly, get the whole package in a short amount of time. But I think it makes sense, especially for these rare diseases, because people, that's it. That's my answer, I guess. Yeah, I think it's worthwhile. They do good work.
Yeah, our time here is almost up. So I just have
wow.
one last question. I know, 17 minutes goes fast. So my last question for you, I think, is just around that. Oh, whoa, a bird just hit my window.
Oh boy.
Is just around that speed question. So you said it took 2 years, and I think that when we hear from patient advocacy groups, they're also really concerned with speed. And do you think it is a viable strategy to start these screens in lower organisms with the idea that they will move?
Yeah, I think it will, because for one reason, it's like, nobody has, a couple of reasons. In terms of time, people don't have all the time on the planet to wait for these things. The second thing is, like, if you do these drug screens in worms or whatnot, if you can find drugs that might be good for just like pure translational repurposing later on, but they're also informative of mechanisms sometimes, right? Because these drugs are also tools for the most part, because they're well known if you're repurposing. So sometimes this gives you clues about the mechanisms of the disease. Yeah, I think it's good to do this stuff in the meantime. There's no sense to wait, like do it linearly. If you can do things in parallel, I think it helps everybody.
That's great. Well, Alex, thank you so much again for joining us on 17 Minutes of Science, and we look forward to seeing more publications from your lab in the future.
Thanks a lot.
Great.
Have a good day.
Bye.